Hello and good morning, students. Today's learning objective is learning objective number 6. Okay, so far we have already done 1 to 5. So we're going to look at objective number 6. Describe and explain changes to the membrane potential of neurons, including resting potential, how resting potential is maintained. Okay, and then the event that occurs during an action potential, and then how resting potential is restored during the refractory period. Okay, so it's quite, this might take about, I mean, if we have to complete all these 3 points, it will take maybe about 1 hour. So we'll see how we can manage to complete the 3 bullet points. Okay, but maybe for a start we looked at resting potential, and maybe if we have time we will look at the events that occur during action potential. Okay, now we're gonna I will send you the soft copy later on after this. So transmission of nerve impulse. Okay, I'm not sure whether you've heard of action potential, but action potentials potential difference, yeah, as well as action potential. So we're gonna use a lot of this term: potential difference, action potential, resting potential, polarized, depolarized, repolarized, hyperpolarized, and so on. Yeah. So we're gonna look at all of these names shortly. Yeah. Okay, so what is potential difference? Those of you who take physics, you might know this, the meaning of potential difference. These are the differences in electrical charge on the inside of a plasma membrane compared to the outside. So differences, okay, differences in the electrical charge, yep, electrical charges. So electrical charges here, we're talking about ions. We're talking about sodium ions, potassium ions, chloride ions, and so on. The differences of electrical charge on the inside of a plasma membrane, okay, compared to outside. Okay. Okay, potential difference always refer to the inside. Okay, it refers to the inside. It tells us what the inside is, or the state of the inside of a membrane. In our case, because we are studying neurons, so we're talking about the inside of a neuron. So what regions are this inside membrane and outside membrane? So the inside membrane refers to the cytoplasm of the cell body and the cytoplasm of the axon. So cytoplasm of the axon is called exoplasm. Okay, so actually cytoplasm and exoplasm, they are the same. Yeah, cytoplasm means the inside of a cell, but the cytoplasm of the axon, is called exoplasm. So they're actually the same. So the inside of a membrane refers to the cytoplasm of the cell body and the cytoplasm of the axon. So this is the meaning of the inside. Whereas the outside of the membrane refers to the tissue fluid. Okay, tissue fluid that surrounds all cells. Right, let's have a look at this one here. The outside of the membrane this is a membrane, so this is actually a neuron. Okay, so this is cell membrane. So inside here is the cytoplasm, or specifically exoplasm, yeah, because this part here are the axon. Okay, axon. So cell membrane, and then the cytoplasm, or specifically exoplasm. Okay, now if we take this as an example, yeah, in an axon, okay, this is normally resting, resting state, meaning when the axon is not doing anything, when the axon or when the neuron is not firing action potential or is not conducting any electrical impulse. Yeah, so when it's resting, so normally when it's resting, the inside of the membrane has less positive ions compared to outside. So inside, less positive ions and more positive ions on the outside, and this is during resting state. Yeah, resting state means when the neuron is not conducting any electrical impulse or is not firing any action potential. So there are less positive ions on the inside compared to the outside. So if you were to measure the potential difference of this, yeah, the potential difference inside compared to outside, then this will give you roughly about minus 70 millivolts. Okay, so if you were to find out the potential difference here, this will give you about minus 70 millivolts. Okay, minus 70 millivolts. So this is the potential difference, meaning the inside is 70 millivolts lower than the outside. Now, so in order to give a symbol, so less positive ions means negative, as you can see here. Less positive ions means negative, and more positive ions means positive. Okay, so this is just a symbol. Okay, it's just a symbol so that we know that negative means less positive ion, and positive means more positive ion. Okay, so negative, when you see a negative symbol, it doesn't mean it has negatively charged ions, yeah? Or the more negatively charged ions, negatively charged ions are present. Although, yes, negatively charged ions are present. For example, things like chloride ions are present, even on the inside as well as on the outside. Okay, so don't worry about the negatively charged ions yet. So just take note that the negative symbol stands for less positive ions, and the positive symbol means more positive ions. Okay, so that's the meaning of that. So this state of less positive ion on the inside and more positive ion on the outside is what you see when the neuron is at resting. Resting means not doing anything, stationary, is not firing any action potential, is not conducting any nerve impulse, the axon is not depolarized, yeah. So it's just basically doing nothing. Okay, and this is what happens. The inside is less positive compared to the outside. So outside is more positive, less the inside is less positive, outside more positive. What it means is that the inside has less positive ions compared to the outside. Okay, so when scientists conducted how did they know that this axon has action potential? Yeah, sorry, it has potential difference. How did these scientists know that exons have potential difference? So what they did was they did experiment on squid. Okay, on squid. So they put one electrodes on the inside and one electrodes on the outside. Okay, just like this one here. Internal electrode, external electrode. So they put one electrode on the outside of the axon squid, the squid of the axon of the squid, and then they put one electron on the inside of the axon of the squid, and so you get this this display on this CRO, cathode ray oscilloscope, and it gives a certain value. Okay, it gives a certain value, and the value is about this is during resting, yeah, resting meaning not doing anything. So when we look here, so it doesn't say here about the resting. Yeah, so basically, as you can see here, so one electrode on the inside, one electrode on the outside. This is the axon, okay, axon of the squid. One electrode on the inside of the axon, one on the outside, and so it gives a certain value here, yeah. And if it's resting, it should give minus 70 millivolt, okay, minus 70 millivolt if it's resting. If it's not conducting any nerve impulse, it should be around minus 70 millivolt. And so there's another term. So the first term that we learned just now is potential difference, okay. So potential difference is basically the differences in the electrical charges inside compared to outside. Okay, and now we look at the resting potential. So the resting potential is the potential difference which exists across the axon membrane when the neuron is not conducting an impulse. So it's not conducting an impulse means it's not firing any action potential. Okay, so what is this action potential? You will learn later on. Okay, so just bear with me. The inside of the membrane is negative compared to outside. Inside of the membrane is negative compared to outside. So it's just like what we have just covered just now here. This is exactly what happens at resting. Yeah, inside is less positive compared to outside. Or if you were to use the symbol, inside is negative, outside is positive. When we say negative, it means less positive ion. When we say positive, more positive ion on the outside. Okay, the axon is at rest when it's not conducting an impulse, and when measured, the potential difference across its membrane, such as axon found in squid, using the above apparatus, gives a value of minus 70 millivolts, or sometimes it can also be slightly lower, minus 65 millivolt. Okay, so in this one here, okay, so when this is, let's say if this axon is from a giant squid, you put an electron inside, you put electrode inside the axon and then outside of the axon, and then it should give a value of around minus 70 millivolts. So it's not conducting anything. It's not conducting any action potential. Okay, so this minus 70 millivolt is what we call the resting potential. Okay, so resting potential, you may have to memorize. It gives around minus 70 millivolt. Or sometimes minus 65 millivolts. I think the conventional, the normal figure is minus 70 millivolt. So the minus sign, what does it mean? This minus when we say that minus 70 millivolt, what does this minus stand for? The minus stands for that the inside is 70 millivolt or 65 millivolt lower. Yeah, lower than the outside. Okay, so this means that the electrical potential on the inside of the giant squid is 70 millivolt lower, meaning outside is higher, the inside is lower. Outside is higher, inside is lower. How low is the inside? It's about 70 millivolt lower. Okay, and in the resting state, the axon is said to be polarized. Okay, resting state, the axon is said to be polarized. So this resting potential and then the axon is said to be polarized. It comes together. Okay, this is like a package. In a resting potential, the axon is said to be polarized. So this resting potential and polarized, this term, they come together. Okay, it's like a package. Resting potential, axon is said to be polarized. Okay, so polarized meaning you should know that minus 70 millivolt. Because there are many terms that you need to learn here. Polarize, depolarize, repolarize, hyperpolarize. Yeah, all these names you have to learn. Okay, so resting potential, axon is said to be polarized. Okay, so you can see some definitions here, some terms. A membrane is said to be polarized if a potential difference is maintained and cause it with the inside being more negative. So polarized means inside is negative compared to outside. So that's the meaning of polarized. Meaning resting potential, yeah? When the axon is in its resting potential, inside is negative, outside is positive. The membrane or the axon is said to be polarized. Okay. Now, depolarized is opposite of polarized. So depolarized means, so if we say that if we have an axon here, inside is negative, outside is positive, we say that this is polarized. Yeah. So depolarized means the opposite, meaning inside is positive, outside is negative. So this is depolarized. Okay, depolarized. And this is only temporary, of course. Temporary, just a short while. Okay, so actually polarized, the opposite of polarized is depolarized, which means that inside is now positive compared to outside. So if you look at the definition here, depolarization is a decrease and then temporary reversal of the potential difference so that the inside, okay, now you see the inside becomes positive with respect to the outside. It's only temporary. Okay, temporary. In discussing potential differences, including resting and action potentials, it is usually more convenient to just refer to the axon and the axon membrane. It should be remembered, however, that these processes can occur all parts of the neuron, plasma membrane, and not just the axon membrane. So when we talk about potential difference, normally we make reference to axon, yeah, or axon membrane. Okay, that's the meaning of this. When we talk about potential difference, normally we say action. We mention about we make reference to axon or axon membrane, but bear in mind that action potential doesn't only occur here, yeah, in the axon or axon membrane, but action potential occurs throughout, yeah, from the cell body. Action potential occurs throughout, yeah. Let me just get clarification here. So basically, it starts from the cell body, yeah, it occurs all regions. Okay, so it's not actually in the axon or axon membrane, yeah. Although when we talk about potential difference, we always, make reference to the axon or axon membrane, but actually in its actual state, it starts from the cell body, yeah, and then it moves all along the parts until it reaches the effector, yeah. Moves all the regions. If it's if it has myelin shield, then it will be slightly different, meaning from the cell body, and then at the nodes of Ranvier, and then here. Okay, because so it will just saltatory. It jumps. Yeah, it jumps. But the point that I'm trying to make is that basically it has to pass all parts of the axon, yeah. It says here, it should be remembered that all these processes occur in all parts of the neuron plasma membrane, not just the axon membrane. Okay. Right, how is this resting potential produced and maintained? So resting potential, so just now it's being measured minus 70 millivolt. So how is this minus 70 millivolt produced, and how is it being maintained? Yeah, how is it produced and how is it being maintained? Now we have to look at the nature of the ions. Okay, so you see that we are still looking at this learning objective, how resting potential is maintained. Yeah? You see the number, the amount of information that I've already given you, so much information coming to this first bullet point, how the resting potential is maintained. Okay, let's talk about some of the ions. The resting potential results from the unequal distribution of charged ions, especially sodium, potassium, these two. Of course, there are also negatively charged ions, which is not it's not really our concern to talk about the negatively charged ions, because it's not in our syllabus. It's not in the A-level syllabus. So let's have a look at this one here. Okay. Look at this one inside and then outside. Okay. So if we look at just sodium and potassium, you can see the total amount inside 450 and outside 480. Okay, and this is exactly you see, inside is 450, outside is 480, and that's why when we draw the axon, we say that this is during resting. Yeah, resting. That's why we write down inside is negative, outside positive. Okay. So outside positive, inside negative. Okay. Because the facts, the facts are stated, yeah. 450 on the inside, 480 on the outside. So less, there are less positively charged ions on the inside, more positive charged ions on the outside. Okay, so this is, of course, don't forget, this is during resting. When they're not conducting anything. Okay, now this table here I've also inserted is also equally important, because now that you are able to see the presence of negatively charged ions. Yeah, this is also important, especially when we talk about the propagation of this, the transmission of the action potential along the axon. Yeah, there are also negatively charged ions. Yeah, like for example, chloride ions and organic anions. Organic anions means these are like, for example, the amino acids which are negatively charged. Yeah, but not mentioned here. Okay, so bear in mind that inside this axon as well as outside of this axon, there are also negatively charged ions such as chloride ions, which are not mentioned, which are not really important in the A-level syllabus. Okay, so when you look at the concentration of the negatively charged ions, there are more negatively charged ions on the outside compared to the inside. Even if you combine, there are more negatively charged ions on the outside compared to the inside. Okay. Right, how is this resting potential produced and maintained? This, so again, so after knowing this, values, yeah, knowing these values, how is this minus 70 millivolt maintained? How is this maintained? Okay, the first one is the effect of the differential permeability. Differential permeability means the permeability of the membrane to potassium and sodium are different. So differential here comes from the word different. Differential permeability, it sounds, when you see the word differential, it sounds complicated. Actually, differential, it means different, meaning the permeability of the membrane or axon membrane to sodium and potassium are different. Okay, this is during resting. So for example here, so we are still talking about resting. Okay, resting. So naturally, there is more potassium on the inside, less on the outside. Okay, we use a different color. And more sodium on the outside and less on the inside. So this is what happens naturally. Okay, naturally. There are more potassium inside and then more sodium outside. More potassium inside compared to outside, and then there is more sodium outside compared to inside. Okay, so how is it possible that, so now we have to ask the question, how is it possible that the inside is negative? Yeah, how is this possible that the inside is negative and then the outside is positive? Okay, when you have this different concentration of potassium and sodium, okay, so it's like this. In the resting state, okay, this potassium ion channel is open. So this potassium ion channel is open. whereas the sodium channels are closed. Yeah, well, it says mostly are closed. Some are open, some are actually open. So these are closed. Okay, so now you see that the potassium will lift. Yeah, so this potassium will leave the inside of the membrane. Yeah, because the channels are open, the potassium channels are open, so more of this potassium will leave. Yeah, will leave the inside. Whereas for the sodium cannot enter. Although some will enter, like out of, it's not entirely closed. Yeah, some are actually open. Yeah, some are actually open, but most of the sodium channels are closed. Yeah, some are open, most of the sodium channels are closed, and so the sodium cannot enter. And because of this scenario, more potassium leave the inside. The sodium cannot enter here, and that's why the inside is more negative. So now the inside is negative. Outside is positive. Okay? So this is reason number one. The reason number one is that the membrane is more permeable to potassium, but not permeable to sodium. Yeah, differential permeability of the membrane to sodium, meaning the membrane has the permeability of the membrane to sodium and potassium is different. So how different? How is it different? We will answer directly that the membrane is not permeable to sodium, but the membrane is more permeable to potassium. Okay, not permeable to sodium, but more permeable to potassium. Okay, not permeable to sodium, more permeable to potassium. So that's why inside is negative compared to outside. Okay, so that's reason number one. So here we say most. Yeah, the term most of the sodium protein channels are closed, and most or more of the potassium channels are open. Most of the sodium channels are closed, and more or most of the potassium channels are open. So meaning there are sodium channels which are open, but then majority of the sodium channels are closed. Okay, that's the meaning of that. Right, this is a very good illustration of what I've just mentioned. As you can see, this one is open. Yeah, this is also open, but then majority is closed. Yeah, the red color, majority is closed. Majority is closed. Likewise, with the potassium, some are closed, but then majority are open. Okay, so if you look here, see the term most. Just want to highlight that the word most. Yeah, so it's wrong to say that they are all completely closed. Okay, so most of the sodium channels are closed. Permeability to sodium is low. Yeah, and then most of the potassium are open. Permeability to potassium is high. Okay, so this is reason number one. You can see that, just the first bullet, we are just looking at the first bullet point here, and it takes us, already about, I think, half an hour. So please bear with me. Right, and that's the first reason. What are we talking about here? We are talking about the maintenance of 70 millivolts. How is this minus 70 millivolts is maintained? How is the inside negative compared to the outside positive? That's what we are trying to address. The question, we're trying to address the question, we're trying to answer the learning objective. Why or how is this minus 70 millivolt is maintained? Why is the inside negative and outside positive? How is this maintained? So the first one is the different permeability, permeability of the membrane to sodium and potassium. Membrane is not permeable to sodium, but the membrane is permeable to potassium. So that's reason number one. The reason number two, which is the last one here, the action of sodium-potassium pump. Okay, sodium-potassium pump. Okay, let's have a look here. Okay, so when we say about sodium-potassium pump, of course, we're talking about active transport. And of course, we know that active transport will require the hydrolysis of ATP. Now, so let me draw here. We say that high potassium inside, low potassium outside, high sodium inside, low sodium, high sodium outside, low sodium inside. Okay, now this, you have to memorize this. High potassium, low potassium outside, high on the inside, high sodium on the outside, low sodium on the inside. You have to memorize this. So when we talk about apart from the potassium channel and then the sodium channel, because of the difference in concentration gradient of potassium, different in concentration gradient here, as well as sodium here. So when we talk about the sodium-potassium pump, okay, so sodium-potassium pump is against its concentration gradient, isn't it? So against its concentration gradient means from so let me redo this again. High sodium, potassium, low potassium, low potassium, high sodium, low sodium, high sodium. So I'm just going to put the sodium potassium pump here. Okay, this is called the sodium. Let me use a different color. Sodium potassium pump. Sodium potassium pump. So it is a pump protein. It is a transmembrane protein. It's called the sodium potassium pump. So what happens here is that it pumps to sodium out. Okay, sorry, three not 2, 3. It pumps 3 sodium out, and it also pumps 2 potassium in. Okay. So this pump pumps 3 sodium out, and at the same time it also pumps 2 potassium in. Yeah, because pumping is always against its concentration gradient. So against from here low to high, pump 3 sodium out, and then this also low to high, it pumps 2 potassium in. Okay, so when you look at the charge, you can see now why it's, you see, 3 remove 3 positive charge and then add in 2 positive charge. Yeah, this is basically a removal of 3 positive and then adding of 2 positive. Yeah, removing 3 positive and then here adding 2 positive. So if what will happen now? It becomes the inside more negative, isn't it? Because it will lead to even less, positively charged in the inside here, because 3 positives are removed and then only 2 positives are being put back, so the inside is even more negative. Yeah, so the inside here is made even more negative and outside will be made more positive. So that's the meaning of the sodium potassium pumped. Okay, so those are the 2 reasons of the resting, how the minus 70 millivolt is maintained. Yeah, so this is the summary of that. Okay, summary of this. So you can see the first one is permeability. Yeah, the first reason is different permeability to sodium and potassium. If I can write down using a different color. The first one is different permeability of axon membrane to potassium and sodium. That's the first reason. Different permeability or differential permeability of axon membrane to potassium and sodium, as we can see here quite clearly. Yeah, you can see most of the potassium will leave, but then the sodium cannot enter. This one cannot enter. The sodium cannot enter because some are open here, but then most of the sodium channels are closed. So if this is the case, then the inside will be more negative, is made negative. Negative means less positive ions. Outside, of course, more positive. So that's reason number 1. Reason number 2 is the action of sodium potassium pumped. Okay, action of sodium potassium pumped. As you can see here, 3 sodium are removed and then 2 potassium are being put in. Yeah, this is removed, 2 potassium are being put in, and so this is the sodium potassium pump. And so now the inside is more negative, is made even more negative compared to outside. Okay, and this is how the minus 70 volt is maintained. This is how the resting potential is maintained. Yeah, why the minus 70 millivolt is obtained during resting potential? Number one, different permeability of the membrane to potassium and sodium, and number two, different permeability of the first one, different permeability of the membrane to potassium and sodium, and the second one is the action of sodium and potassium pumped. Okay, this is taken from different textbook. You can, there's nothing wrong looking at the process of this. Don't forget the process. The process. This is facilitated diffusion. Yeah, this one here is facilitated diffusion. Facilitated diffusion. Yeah, this is of course this is active transport. You should know this already by now. Okay. So it's stated here, facilitated diffusion and active transport. And don't forget, when it is actively transporting, ATP will be hydrolyzed, okay, to provide the energy for the pumping. Same thing here as well. ATP is hydrolyzed during the pumping. Okay. You can see again, ATP is hydrolyzed during the pumping. Okay, and just for you to know that there are about 100 to 200 sodium potassium pump per square micrometer. I mean, that's quite a lot, yeah, per square micrometer. There are about 100 200 sodium potassium pumps, so you know that they're actually working very, very hard to maintain the minus 70 millivolt. Yeah, the inside is 70 millivolt lower than the outside. That's the meaning of the term minus. The minus 70 volt means that the inside is 70 millivolt lower compared to the outside. Okay, so just some other terms, the negatively charged ions like the chloride ions are also actually involved, but this is not in the syllabus. Yeah, it says here it's not essential in the in A level. But don't forget that the chloride ions play a very, very important role when we talk about the local electrical circuit, the setting up of the local electrical circuit. It's very, very important when we talk about we will be talking about these chloride ions when we mention about the propagation of action potential along axons. It says here, in addition to sodium and chloride, in addition to sodium and potassium, the chloride ions are present on either side of the neuron membrane. These are involved in nerve impulse. You see, these are involved. It says they are also involved in nerve impulse. For example, the organic protein ions are negatively charged, are only found inside the membrane. The membrane is impermeable to these ions, which therefore remain inside and contribute as well to the resting potential. Okay, so when you enter uni, you will learn this in more detail, but in A level, it's only enough with sodium and chloride. But the chloride ions will also be mentioned again, especially in the propagation of nerve impulse. Yeah, so I think that's all for today because it's taking too long already. Now the action potential, this one will cover this tomorrow. We take it slow and steady. Yeah, so just for today is only the first bullet point, how the resting potential is maintained. All right, that's all for today.